Perfect. Thank you, Andy. And thanks to both Andy and Lindsay for their leadership of the Continuum Scholars Program this summer, as well as all of the other faculty who played a part in this as the Continuum Scholars. It was great. We had really fruitful conversations and time to reflect on things that I hadn't before during my time at Hope, at least not with others. So I will be talking a little bit today about my work with the mitochondria. I love this image that's on the slide right here. All the green lines there are mitochondria within a cell, and they kind of form this like energetic life force that we'll talk about a little bit more as we go on. So today what I hope to talk about, what are the mitochondria and why am I excited about studying mitochondrial function? So I'll give you a little taste of that. Then I'll tell a short snippet of ongoing research in my lab that's focused on the regulation of mitochondrial DNA transcription. And then finally end with a discussion of my faith and my scholarship. So first, what are mitochondria? And so as we've already covered, mitochondria are the powerhouse of the cell. And so again, from kind of any introductory biology class, even as far back as probably middle school, we learn about the different compartments of a cell. And I can remember like putting cell models together and putting the little pieces and parts with styrofoam or something. And I always kind of love the mitochondria. We think of it as this little bean-shaped thing that you see on that mug. Remember this mug. I'm going to talk about this mug later in the talk, but we'll go from there. So what are mitochondria? We can also answer this question. Star Wars has a great answer to what are mitochondria. So I'm going to show a quick clip here that will help us to understand what mitochondria are.
Master, what are midichlorians?
Midichlorians are a microscopic life form that resides within all living cells.
They live inside me.
Inside your cells, yes. And we are symbionts with them.
Symbionts?
Life forms living together for mutual advantage. Without the midichlorians, life could not exist, and we would have no knowledge of the Force. They continually speak to us, telling us the will of the Force.
The Force?
The Force is what gives the Jedi his power. It's an energy field created by all living things. It surrounds us.
Midichlorians are a microscopic, but this is going to be a problem if this keeps happening. So let's see here. Is that better? Okay, maybe it was just twisted a little bit. Okay, so the quote there is, midichlorians are a microscopic life form that reside inside of our living cells. Without the midichlorians, life could not exist, and they also explain the Force. Okay, so that's all I have to say today. That's why I study the mitochondria. No, I'm joking, right? So this is a funny little clip that I'm not the only mitochondrial biochemist or biologist who likes to show this thing. It's kind of a joke. But there's a lot of things that are kind of true in here, right? Like life couldn't exist without mitochondria. They are sort of a microscopic life form that reside inside our cells through this process of symbiosis. And so I'm going to hit on all of those things as we go forward. But let's dig in a little bit deeper. What are the mitochondria? So the powerhouse, what does that mean that mitochondria are the powerhouse of the cell? Well, mitochondria are required to generate most of this molecule called adenosine triphosphate, or ATP. We'll ignore the chemical structure and just focus kind of on these three circles, which are the phosphates of that molecule. And we can think of ATP as running around the cell as like a charged battery. And it helps reactions occur that normally energetically wouldn't be favorable. So the hydrolysis of ATP, or the breakdown to ADP, now we have two phosphates here, is often coupled with a large number of different metabolic reactions that, again, could not occur unless we use ATP with it. Now, where do the mitochondria come back in? Well, the mitochondria come in that they regenerate this ATP. So they take the dead battery, ADP, that's not really useful for anything, and they synthesize a ton of ATP. And so without this, a number of metabolic reactions couldn't happen. Life as we know it in our cells couldn't occur because we wouldn't have ATP, and we need the mitochondria to generate this energy currency, which is why they're known as the powerhouse of the cell. Just some fun facts here. About 110 pounds of ATP are converted to ADP daily. This is a ton, right? That doesn't mean a 110-pound person is all ATP. what's happening here is that each ATP equivalent is recycled more than 500 times each day. Okay, so the mitochondria have to be there to recycle ADP to ATP, and this process is occurring rapidly and all of the time. And so that's why the mitochondria get their name as the powerhouse of the cell. There's some other fun facts about the mitochondria, if I haven't sold you on them yet with Star Wars and the powerhouse. There are 10 million billion mitochondria in a human. That is approximately 10% of our body weight. Mitochondrial proteins, I'm a protein biochemist. I get excited about studying the machines of our cells, the proteins within our cells that do kind of all of this work that we need them to do, account for about 7 to 10% of the entire human proteome. And then finally, the mitochondria are central to metabolism of all sorts of dietary fuels. So the mitochondria don't just make ATP. They do a whole ton of other things beyond even just these, beyond just metabolism. And when I think about my interest in the mitochondria and how I got to studying the mitochondria, I think it begins with metabolism and this interest in metabolism. I remember being a kid and really being interested in maps and puzzles and logic and how does this all work together. And when we think about metabolism, so this is a metabolic map. It's not intended for you to be able to read the pieces, but just that you can see, right, all of these different chemical conversions. So each kind of line here is some chemical reaction that's occurring within our cell that's involved in metabolism. And I think it's beautiful that all of these things happen in our cells. It's regulated, it's complex, and trying to understand how all of this works together is something that really interests me. And the mitochondria, which if we look just kind of at this dark gray box, all of these metabolic reactions are occurring within the mitochondria. They're really central hubs to cellular metabolism. Additionally, another reason why I'm excited about the mitochondria is this idea about their importance in human health and disease. I think I've heard a number of colleagues share a similar story that they entered into undergrad thinking that they were going to go into medical school. They were interested in science and they wanted to help people, and med school was the way that you would do that. And I was there. I was there probably until the end of my junior year. I was pretty convinced I was going to med school. And some things happened that I said, oh no, that's not what I want to do. And so I transitioned into realizing you could use science as a researcher to also impact human health and to understand human disease. So again, I used to say to students, tell me a disease and I'll tell you how the mitochondria is implicated. Now, that's hubris, right? It's overstatement. But there's a little bit of truth that as mitochondria begin to fail, we see these types of things in aging. If we think of them as engines, and as your engine becomes less efficient, right, you release heat. That's bad for a car. Similarly, that's bad for a person as we get less efficient mitochondrial function. So mitochondrial dysfunction has been implicated in diseases like cancer, neurodegenerative disorders, specific genetic kind of mutations within the mitochondrial genome that I'll talk about that then cause disease. And so metabolism gets me excited about the mitochondria, and their roles in disease also gets me excited about the mitochondria. So the goal of my lab at Hope is to understand basic biochemistry of mitochondrial function in order to enable a deeper understanding of mitochondrial dysfunction that occurs in human disease. So we don't come at it from a disease perspective. I don't say our lab studies cancer. I say our lab wants to understand better how the mitochondria work in the hopes that someone else might be able to use that information in the context of a human disease. And everything that we do is focused on the mitochondrial genome, or mitochondrial DNA, which is shown in this picture. So again, we see our little bean of a mitochondria, and within each mitochondria, there are a number of copies of mitochondrial DNA. And where you might be most familiar with mitochondrial DNA is through lineage tracing, because it's thought to be maternally inherited. And so some people can, you can trace mitochondrial DNA throughout kind of maternal lineages, perhaps even to some mitochondrial Eve, shall we say. But I'm excited about mitochondrial DNA for some other reasons that I'll tell you about. Mitochondrial DNA, again, the small circular genome. Recall we have genetic information now in two places, in the mitochondria as well as in the nucleus, right, where we think of the bulk of our DNA. Before I get too far, I know that this audience, we have people from all different places, so I want to go through some words that I'm going to use and make sure we all have the same kind of background. So, a central dogma of biology, we start with DNA, so either mitochondrial DNA or DNA in our nucleus. We can think of this DNA as like the blueprint to an entire house. Through the process of transcription, that's a word I'll use a lot in this talk because we study transcription. Through the process of transcription, we make what are called messenger RNAs or mRNAs. So that's like saying, hey, we want to build the door of the house. We don't need all of the instructions in the DNA. We just need the instructions for the door only. So we pull the instructions from the door only and make a message of that. Through the process of translation, we go from mRNA and we synthesize our proteins. So much like we could take the blueprint for the door and then we could build the door. Okay, so that's kind of these words that I'll use and talk about. So what do we study in the lab? What are we thinking about? So we start up here with a mammalian cell, nucleus and mitochondria. Of course, there are other bits and pieces of that cell, but we're focused right now on just the nucleus and the mitochondria. And your cell could be likened to a city, where all these different parts have to work together in order for appropriate function to occur. Let's focus first on the mitochondrial genome. The mitochondrial genome codes just for 13 proteins. And those 13 proteins are required for cellular energy production, much like a power plant would be required for energy production and distribution in a city. Now, if we compare that to the nuclear genome carries the genetic information for over 20,000 proteins. So this is orders of magnitude greater when we compare the mitochondrial genome to the nuclear genome. And this directs cell function, right? Much like City Hall directs the city function. City Hall might be governed by a city council. The nuclear genome is controlled by transcription factors, among many other things. Now, what I find most interesting when we think about the nuclear genome and the mitochondrial genome is that there are a large number of proteins, 1,500 to 2,000 proteins, of which the genetic information is carried within the nucleus, but those proteins must be transcribed and translated into proteins and then shipped into the mitochondria in order for them to carry out their function. Some of these proteins include transcription factors for mitochondrial DNA, so these things that control the transcription, DNA to mRNA. Also, the protein machinery that's required for transcription of mitochondrial DNA. So, without the nucleus, the mitochondria cannot transcribe its own mitochondrial DNA. It's useless. The mitochondrial DNA is useless inside of the mitochondria unless the nucleus is there to help it. And I think that that's really interesting. And then finally, we talked about the powerhouse and the synthesis of ATP. So there's 90 protein puzzle pieces that are required in order to generate ATP directly. 13 of those are encoded by the mitochondrial genome. The remainder are encoded by the nuclear genome. So the mitochondria on its own cannot even make ATP. It relies on other protein subunits that the information is carried in the nuclear DNA in order for that to work. So this sets up a situation where these two genomes must communicate with each other in order to match kind of energetic needs. The cell says, hey, I need more ATP. The mitochondria can't go off and do its own thing without kind of the nucleus being there to potentially help as well. So the big questions in my lab are, how do the mitochondria and nucleus communicate to coordinate their transcriptional response to changes in cellular energy demands? So again, if this idea, we need more energy, we need more ATP, kind of long term, well, we need to boost the transcriptional machinery potentially, which requires a nucleus. We might need to boost the number of components of the energy production machinery, again, relying on the nucleus. How are these signals relayed between the two compartments of the cell? That's the big picture question that kind of drives my research and my interest. The second question is, how is mitochondrial DNA transcription regulated in human cells? And the third question, which kind of tries to address questions 1 and 2, are that a number of proteins, about 60, are known to associate with mitochondrial DNA, but their roles in transcription and transcriptional regulation are unknown. So my hope during my career is to take a stab, maybe not at all 60, but to start plugging away at these proteins known to associate with mitochondrial DNA and say, are they doing anything important in both protecting potentially the genome or regulating transcription of the genome? So what's our hypothesis? What are we studying in the lab directly? So our hypothesis is that things called protein post-translational modifications regulate mitochondrial DNA transcription. So I presented this just to you a few slides ago, the central dogma. There's another layer of complexity on top of that central dogma. And those are these things called post-translational modifications, where we can decorate the amino acids, the chemicals that are linked together in order to form proteins. We can decorate them with different chemical modifications, and that changes the function of that protein. So for example, we could add a mail slot to the door, or we could put windows on the door, or we could take the doorknob away, right? So we've changed the function by changing the chemical makeup of that protein. So what do these look like? I got to show a few structures here. So one of the post-translational modifications we think about is phosphorylation. So phosphorylation occurs on hydroxyl groups or alcohol groups on 3 different amino acids within our proteins. And we can see chemically, right, that this is different. So we've gone from neutral to negative charge, bigger kind of molecule on there. So we can envision how sticking that in potentially to a protein that binds to mitochondrial DNA, we've now stuck this negative charge in there and made it a little bigger that could disrupt some interaction or cause an impact. Importantly, these are reversible, which is kind of cool, right? So a protein puts this modification on, and a protein can take it off. So it can act like a switch, an on-off switch for protein function. Another modification that we think about is this modification called acetylation. And so again, we're focused on the change that occurs to this NH3 group. It's often positively charged. We've lost this positive charge and again kind of changed the structure of that amino acid. It turns out that mitochondrial proteins have a ton of amino acids that are modified by these different types of chemical groups. So about 2,500 acetylated lysine. So a lysine is the amino acid, and I'll use the word site to refer to these modified places on proteins. 1,000 phosphorylation sites, and they're involved in all sorts of different processes within the mitochondria. And so my work back in grad school kind of started with trying to explore these different modification sites. That really kind of launched me into this interest in mitochondrial DNA because I saw lots of modifications on proteins that were involved in mitochondrial DNA transcription and maintenance. Further, about 5%, maybe less, have a characterized biochemical function. And so while we're really interested in the lab in understanding mitochondrial DNA transcription, this is also helping to understand kind of the bigger picture of why are these modifications within the mitochondria. So I'll tell you about 3 of our favorite proteins that we're focused on. So this is a protein crystal structure. I think they're quite beautiful. I like to look at them. Another reason why I like protein biochemistry. What we're looking at here in the orange and the blue and the green, that's DNA. And these 3 proteins, POL-RMT, which is the polymerase, it's the enzyme that actually synthesizes the mRNA. In yellow here, we see a transcription factor. This transcription factor is important to get the polymerase at the right spot on the mitochondrial DNA. And then the red protein here is TFB2M, or transcription factor B2. And it's super important, and again, kind of this appropriate alignment, as well as opening the DNA up. That's what promoter melting means. We have to open the DNA up in order for the polymerase to get in there and start doing its job. So a lot of our work has focused on TFB2M, although each of these proteins have documented modification sites. And this is either from literature or from some work that we've done in the lab with the mass spectrometer that we have that can kind of identify where on the protein these modifications exist. If you look closely, you see kind of these space-filling balls on TFB2M. Those are 3 modification sites that we've studied and sought to understand their function. I'm going to change colors here a little bit. I like this image a little bit better in order to see these sites. But S197, that means serine 197, so one of those amino acids in that protein, which is known to be phosphorylated. And then we have threonine 313 and threonine 184. So 3 amino acids, all known to be phosphorylated. Nobody knew what they were doing, if they played any important role in protein function. This S197 we looked at, so I often have students look at these pictures and I say, make a hypothesis. Tell me what you think could be going on if the chemical structure of that amino acid changes. And so it's really great kind of thing to look at and kind of investigative work we can do before even doing any wet lab experiments in the lab. So how do we study this? What we do is we use E. coli. We trick E. coli into becoming little protein synthesis machines. So much like an organic chemist can synthesize an organic molecule and purify it, we can have E. coli synthesize a big organic molecule, a big protein, and we can purify it away. That's great, but we have to somehow mimic this phosphorylated group. So here's just one example. Here we see the amino acid serine with it in its phosphorylated form. Unfortunately, we can't easily just make a protein in the lab that's phosphorylated at a single site. So we can't do that. We have to mimic that phosphorylation in some other way. So we can mimic it by changing the amino acid at the position to either an aspartate or a glutamic acid. And you can see now that those kind of look a little more similar than just an OH group, okay? And so we use these, what we call modification mimics, to purify libraries of proteins that are modified, and then we go ahead and we study those in a couple of different ways. So once we purify our proteins of interest, we can look at transcription inside of a test tube. So we can actually say, how well does this protein transcribe DNA? And we can also assess its ability to bind to mitochondrial DNA with the idea that if it can't bind to DNA, it probably can't do its job in transcription as well. And so what we found from this work when we made these phosphorylation mimics of this protein is that when TFP2M was phosphorylated, it didn't bind to mitochondrial DNA as well, and we got a loss of mitochondrial DNA transcription. So we pull TFP2M away, and we no longer can transcribe DNA. I have one data slide, so this is it. So I'm going to show you, so you got to bear with me, okay? So here's just some of the data that support that picture that I showed you on the last slide. So we have a template of DNA. We give it some mitochondrial DNA in a test tube. We give the system mitochondrial DNA. We give it our TFP2M protein, the polymerase, and we say, are you doing transcription or are you not doing transcription? And so we can do these experiments. We'll just focus here on some of the things I have in boxes. So what this is called a runoff experiment. And so that means, can the polymerase start at the initiation site for transcription, and can it run off the template that we give it? And so if we see dark dots, it means, yes, indeed, it did run off. And so, for example, wild type WT here, we see 2 dark dots. That means it did really well. For serine 197, we see 2 dark dots. That means it did really well. For these 2 other mutants, phosphorylation mimics, we see no dots, right? So it didn't do as well. And so those are the data that kind of tell us that when we're looking at T184E, this phosphorylation site, or the phosphorylation site at 3-adenine 313, we don't get productive transcription. We can back up these claims with some binding data. So there's a lot going on here. I'm not going to talk about it all. I want you to focus on just a few things here. So if the protein doesn't bind to mitochondrial DNA, we would say that transcription likely will not occur. And indeed, that's what we found in this study, where transcription is not occurring. And we can look at how well each of the proteins binds to mitochondrial DNA using a method called fluorescence polarization. The higher the number, the worse the binding. So we see high number here, 455 nanomolar, high number here compared to our wild type protein. And so all of this kind of nicely works together to say that we don't have binding and we don't have transcription. So what's next? Well, what's next? I showed you those other 2 kind of protein friends that are absolutely required for transcription. They have a bunch of modification sites. We're kind of systematically working through those proteins, doing very similar experiments to what we showed here to say the core transcriptional machinery, those 3 proteins, what are post-translational modifications doing? What's next? So mitochondrial DNA, again, binds to all these proteins, right? And I said, I'd love to know what all 60 of these proteins are doing in transcription. So we know that there's proteins that are involved in packaging mitochondrial DNA. They're called kind of nucleoid proteins. It involves the core transcriptional machinery, which we're already studying, and a number of really cool metabolic enzymes. And so we're moving on, well, not moving on, but moving into studying some metabolic enzymes as well as we finish kind of understanding the transcriptional machinery, the core transcriptional machinery. With the idea that these metabolic proteins are acting as sensors or rheostats of metabolic state, and then conferring that information to the mitochondrial genome to either enhance transcription or to decrease transcription. Ooh, ooh. Okay, I won't even touch it. All right, so that kind of ends, the little research portion. We'll move on into the third portion here. Maybe we will, maybe we won't, we'll see. I don't know if we have a bad HDMI or what. Okay, well, it could be, we're just gonna leave it because it's working. So we're returning to the mug, right? Okay, and Andy kind of talked to you a little bit about this in the vocational biography that he presented at the beginning. But I entered the continuum program thinking, man, I really want to find this explicit connection between my faith and my scholarship. And I really wanted that. I looked to lots of great people at Hope doing research and can see kind of those explicit connections and thought, that's what it means to be a Christian scholar. There must be some explicit connection that I haven't yet found for my work. So I was frustrated the first 2 days of continuum because I'm like, this isn't helping. I'm not finding anything. And then we read something, and a student stopped by and gave me this mug with a nice little note. And this student wasn't the best student. It was actually a student who took Biochem 1 twice with me. I don't often think of getting gifts from students who've had to take my class twice. And this student, the second time, I was just a cheerleader. I wouldn't say that I went out of my way to do anything extra except to really acknowledge when they were succeeding. And I think that helped that student, and the student did fine and then ended up electing to take Biochem 2 with me. And so that really spoke a lot. And then the student came back a year later and gave me this mug and told me they were off to a master's program and stuff like that. And kind of that, along with some of the readings, kind of came up at the same time. And I thought, maybe it doesn't have to be explicit. And maybe through mentoring and the relationships that I have with students and my colleagues, I can demonstrate my faith in this way, which is kind of defined as purely vocational Christian scholarship. And so I think that through mentoring, I can really demonstrate my faith. I can interact with people in ways that kind of reveal to them things about my Christianity, and it's not always in an explicit manner. So again, mentoring. These are some of the awesome students I've had the privilege of mentoring during my 6 years at Hope in my lab. These are the students that I form the closest relationships with and get to have all sorts of interesting conversations with, whether it's about science or life or whatever. And I feel very fortunate that these students are almost an extension of my family, and I still keep connected with my very first research student. I was invited to her wedding in the fall. And so those are types of relationships that I think being at a place like Hope, being a Christian scholar, kind of afford these types of long-term connections with students. And I hope that through my mentoring, I'm also kind of getting them to the places where they hope to be. So mentoring is certainly one aspect of my vocation and my work as a Christian scholar. But I do think that in the background, I have questions that that kind of are related to origin of life questions. And the mitochondria play a very critical role in life, right? If we think back to the Star Wars video, right, they are kind of required for life as we know it. And so I think as a Christian, I'm very interested in this idea of both kind of science and faith and how they work together. And I think that stems back to the origin of life, right? Like, where did this happen? How did it happen? These types of questions. And so I'll talk a little more science here for a second. So here we see an image of a cell. Very, very, right, early in evolutionary time, some event occurred in which a bacterial cell, a proteobacterium, was engulfed by some sort of early eukaryotic cell. So our cells are called eukaryotic cells. Human cells are eukaryotic cells. So this proteobacterium kind of was co-opted or engulfed by this early eukaryotic cell, and over the course of evolutionary time, we get to the cellular ancestor of kind of our cells. Without this event, without kind of this symbiotic event of the engulfment of this early mitochondrial precursor, life as we know it probably would not exist, right? Because we need the ATP that these things provide in order for the cell to continue to grow and to become more complex, et cetera, et cetera. And so I think in the back of my mind, this is really interesting and something that I like to think about, even though I don't explicitly study this connection. And I'll just take this a little bit of step further to try to explain. So again, at one point we had this ancient bacterial-like cell, and over the course of evolution, we got to our mature mitochondria that we know of today. Now, over this evolutionary timescale, lots of things happened. And so some of the things that happened was that information was transferred to the nucleus. So at one point, this was free living, and it did all of the things that it needed to do on its own. But I already emphasized the point to you today that the mitochondria in our cells now cannot function independent of the nucleus. It relies on this information within the nuclear genome in order to function. So these proteins that are encoded by the nucleus and carried there. But again, throughout evolutionary timescales, we're talking billions of years, the mitochondria also retained this small genome. Why? Why did it want to retain this small genome? And that, to me, is absolutely fascinating, that the mitochondria has this genome that encodes only 13 proteins, but it gave everything else up to the nucleus. If I knew how to study that, I would, but I don't. But I think that that idea is kind of really interesting, and again, brings in this perspective of faith and origin of life that kind of drives an interest in the mitochondria. And so I'll just end with a few quotes. The more I study nature, the more I stand amazed at the work of the Creator. And so I hope that today I've given you a little bit of a sense of that through my work and through my scholarship, I stand kind of amazed at that all of this works. How does it work, right? How does metabolism all work? It's so complex. How did this mitochondria come to be? And why did these things get kept in the mitochondria where other things got shipped away? And so this is a quote by Louis Pasteur, which another kind of interesting connection here. Louis Pasteur was one of the first scientists who figured out that if there was oxygen around, things grew better. And so that's all about the mitochondria. Mitochondria rely on oxygen to generate ATP. And I'll end just kind of with a few more things about my thoughts on science and faith. And so there are people who are much more articulate about this than I am, and so I'm stealing from them for this presentation. So science and faith is always this thing, right? And we can think about that when we think about origin questions. We can think about when we think about evolution questions. And it seems like sometimes those things are at odds with each other. And I left probably undergrad thinking those two things were at odds with each other. And I didn't know how to reconcile it for myself. I still don't, but I'm kind of comfortable with that. But I remember turning to some books, and one book that really helped me kind of put these pieces together about, like, I can be a scientist, and I can also be faithful. Francis Collins, you may know him. He just up until last month was the director of the NIH. He certainly has become much more in the public scene with COVID and things like that. He was the leader of the Human Genome Project, the project that sequenced all of the DNA within a human. And he writes in this book, "The God of the Bible is also the God of the genome. He can be worshipped in the cathedral or in the laboratory. His creation is majestic, awesome, intricate, and beautiful, and it cannot be at war with itself." And so I resonate strongly with this kind of quote and just kind of stand in awe of what's happening in our cells at an atomic level. He further says in another quote, "It's a miracle that it, speaking of the universe, has order, fine-tuning that allows the possibility of complexity and the laws that follow precise mathematical formulas. Contemplating this, an open-minded observer is almost forced to conclude that there must be a mind behind all of this, a profound truth that lies outside of scientific explanation. And so I've used the puzzle analogy already, and for me, I think there's lots of puzzle pieces that we as scientists can put together. There's probably always going to be like that piece that slid way under the couch or went down, and my daughter loses puzzle pieces all the time, that we can't put the puzzle all together. And so to me, that says there's something greater, that there's a greater mind behind all of this that kind of has helped this to occur. So with that, I will end, and I'll just say a quick acknowledgement to all of my lab members, folks listed there, some collaborators, funding for the projects, and then again to the Continuum Scholars Program and to Hope College for this experience.
Thank you, Kristen. Thank you for providing presentations so wonderfully accessible to those of us who for Lakeview.
Yes.
Love it. But friends, we have time for questions, insights, interactions with this very, very thoughtful gift that Kristen's given us today. Who would like to jump in? I will. Sometimes it works and sometimes it doesn't. Let me unmute it. Would you like to go first, Laura, or are you just telling me to do that? I'm just telling you to use my. Okay.
You should be good.
So I can understand that evolution works very well on DNA in the nucleus. How does it work on DNA in the mitochondria if it's not doing the whole meiosis thing like it does in the nucleus, or does it? Does that question make sense?
I think so. I don't know. I think so. So you're saying over time, evolutionary time, our DNA has been altered within our nucleus. What happened in the mitochondria?
Right. Because I can, if cellular information, right, in the DNA moves to the nucleus, then it there can use sexual and other mechanisms to let evolution select on it and improve etsfunction.
Yeah.
Does that same mechanism occur in the mitochondria? Is there meiosis and recombination there?
No, there aren't. I have never thought about that question. That's a very interesting question. No, those types of things do not occur in the mitochondrial genome. And I don't know the answer to your question. So I'll try to answer it, but it might not be perfect. So that's what I'll say. And I don't know a ton about this. But so the mitochondrial genome, again, is inherited by the mom through through the maternal side. And it's there are multiple copies of the mitochondrial DNA within even a single mitochondria. In development, there's what's known as the mitochondrial bottleneck, where the amount of mitochondrial DNA actually kind of shrinks. We'll go with that. The amount of mitochondrial DNA is actually limited, and then it's also just from the mom. So we only want kind of the best mitochondrial DNA to then go on in the process of development. And so that in some way kind of limits what happens, at least kind of in sexual reproduction, about how the mitochondria are inherited and things like that. But no, the other events that you're talking about don't happen. The mitochondrial DNA is kind of susceptible to damage that occurs within the mitochondria. But again, we try to limit that with limiting the amount of mitochondrial DNA before kind of things take off. That's the best I can do.
Okay.
I think.
Thank you. It certainly makes sense that with all the high energy stuff going on in the mitochondria, you want to keep sensitive things outside.
Yes.
Thank you.
Yeah.
I have a much simpler question.
Okay, great.
But first, let me thank you for your presentation. The clear metaphors were really helpful to somebody like me. My name is Dave, and I'm in the psych department. But long ago, in the early 1960s, I was a chemistry major, and I was a biology minor. Unfortunately, I've forgotten most of what I learned, although I've stayed with me. What I've learned is my scientific empirical orientation to life. But I don't recall learning about mitochondria. When did we come to appreciate mitochondria and their importance?
My goodness. That's definitely not an easier question. I should know the answer to that, but I don't know. Early 1900s? I'm looking at Leah. I don't know. Do you know? Yeah, I don't know.
How long ago were you in school, Dave?
I would definitely say people knew about the mitochondria before then.
70s, yeah.
Yeah, I mean, I think that a lot of details probably about the mitochondria. I'm going to say 60 to 70s is like the thinking about electron transport chain and ATP synthesis, like those details. But Linus Pauling is certainly way before that, and he kind of was one of the first to start to think about these things. But yeah, that's a good question. I don't know those dates. Yeah, Jason. 1857. Okay.
Thanks, Kristen. That was really great. My question will belie my biological ignorance because I was even younger than David. It wasn't longer ago, but I was younger. I was 13 the last time I had any of this kind of content. Are there still eukaryotes that don't have mitochondria, or that it was, once we have mitochondria, all eukaryotes now have them?
Yes, correct. That event occurred early enough in evolution that, fungi. And
so having them wins out over not having them.
Yes.
Okay, so you said way long ago bacteria was engulfed by the cell, which is now the mitochondria, but now the mitochondria is interdependent with the nucleus in order to do anything. How much has the mitochondria really changed then, throughout that evolution, to the point where now it almost relies on the nucleus to be able to do what it once did on its own?
Yeah, that's a really great question, and I don't have a great answer to that question because I don't think anyone really knows what that early proteobacterium exactly was. Maybe people know more than what I'm saying, but certainly lots of genetic information changed, lots of regulation changed, right? The mitochondria actually have probably developed more functions than what they had when they were kind of on their own. And we're talking billions of years here of things that can happen and occur. So I don't know the explicit, this pathway now the mitochondria has and it didn't. But I would envision that there's a lot of things happening within the mitochondria that didn't happen just in that bug. I would say that bug probably made a lot of ATP, right? And then things kind of went from there. Mm-hmm.
I have a question about, I know you study how mitochondria work and not how they don't work. But I know that in Dance Marathon coming up in a month or so, we've got at least one, maybe more kids that have a mitochondrial dysfunction, which means sometimes it doesn't work out right. And I'm thinking, I know very little, again, ignorance. I'm Laura. I have no academic affiliation. I'm staff. So very ignorant on all of this. But how does, if it is coming only from the mother, and I think of recessive and genetic traits and, the very basics that you learn in middle school, how does a mitochondrial disorder appear? Is it a new mutation within the mitochondrial DNA, or is it how the mitochondria interact with something else? Do you know anything about disease-causing mitochondrial dysfunction?
Yeah, so most of the diseases that children are afflicted by that we're probably thinking about in this case, not in the context of a thing like a cancer, a neurodegenerative disorder, which is kind of a long-term decline in mitochondrial function. Those disorders are often point mutations, single nucleotide mutations within the mitochondrial DNA that then impact energy production. So commonly children will have kind of brain issues in tissues that use a lot of energy, brain, heart, skeletal muscle. We see issues with children, it comes out in children. So those mutations can be inherited through the mom. Or they can be kind of mutations that occur sporadically. All mutations are random. So a random event that occurs and it's in a really bad spot, right? And that really bad spot then causes the disease. But it can be inherited. Now, there are other diseases that are caused by the other things that you mentioned there, but the ones that we see in children are how I just explained.
I have a question, and that is maybe kind of tagging off of this. What are the implications of mitochondrial transcription research going forward? In other words, as you do this research, is it simply to advance the biological science? Are there other implications for disease or that kind of thing as you do this research?
Yeah, that's a really great question. So I think my goal really is let's understand how the mitochondria function better, right? We don't know these pieces of the puzzle. Let's try to understand them. And through understanding the transcription, perhaps we can then understand further how the cell works, right? A bigger question of biology. There are some human disorders that are caused by defective mitochondrial DNA transcription, kind of just these random energy issues. We looked at a paper this summer with the mitochondrial polymerase having some mutations and issues that led to its dysfunction. And so there are some one-off cases where we see these types of things in human disease. And so there are implications there, but really our root is to try to understand basic biochemistry that helps us understand how a cell functions.
Oh, one more here. Can I? Yes. Let me.
Thanks. I don't really have a question. I just wanted to comment. I appreciated your discussion of how you combine your faith with your teaching. So I'm not a biochemist, but I do teach a class to kinesiology students who don't have a lot of chemistry. It's called Regulation of Human Metabolism, and we learn about all of the pathways and how we're able to provide ATP for sprinting compared to, prolonged exercise. And one of the ways that I try to combine my faith with the concepts that we talk about is all the intricate ways that the different enzymes and the pathways work together, and ask the students constantly, why does it make sense that it works this way to allow this pathway to work, whereas that pathway doesn't work when this one is working? And I think the students appreciate that. So we don't go into any of the details with structures or really bonding or anything with the students, but I think just thinking of the big picture of how God designed our bodies to work the way they do is a nice way to do it. So, and then Dave, I think when I teach about this, it was 1967 that the mitochondria, we learned about the electrochemical gradient and how that allows oxygen to be used. So maybe that's why you didn't learn that so much in the 60s. Anyways.
Thank you.
Dr. Meyer, would you like to have the last question here?
Again, it's kind of a stupid, naive question, but is there a boundary between biochemistry and microbiology? And if so, what is it, and which side of that boundary are you on?
Yeah, I think as scientists we start to use these terms a little bit loosely, but my definition would be a microbiologist studies bacteria and yeast and things like that, whereas as a biochemist, biochemistry is probably the biggest umbrella where people take that into different places. But as a biochemist, one might be a protein biochemist like what I am. I study protein function, molecular interactions, functions of molecules within our cells, those types of things. So that's how I would loosely define those two things.
Well, I want to tell you again, Kristen, thank you for a fantastic lecture today. It was a delight to have you be a part of our group this summer. Your thoughtful reflection here on your own scholarship and your faith was characteristic of conversations we had this summer, and I just really appreciate the time with you and you sharing with us today. Can we thank Dr. Ditton-Huffenried again, please?
Thank you.